Hi, I'm Judy Cole, the Executive Vice President and CEO of the MIT Alumni Association, and I'm delighted to welcome you to this web production of the MIT Alumni Association.
Hello. Welcome to the MIT Faculty Forum Alumni Edition. I'm Kathy Clabby, a 2008 Knight Science Journalism Fellow from MIT. I'm a freelance reporter and an environmental health reporter for North Carolina Health News. I'm going to serve as moderator for our discussion today on advances in autism research, an incredibly important topic that an increasing number of people want to better understand, especially families. This is an interactive forum, so we encourage your questions. Use the Google form below, please, below the live stream to ask questions, or tweet one to hashtag MIT Better World. We'll get to as many questions as we can. We are joined by 4 scientists involved in autism research, mostly on the molecular level. They're working to better understand the biological roots of neurological differences that are associated with the autism spectrum. Difficult work. They are Susanna Barrett Mierau of the University of Cambridge, Jean Yo, the University of California at San Diego, Neville Sanjana. Neville, I hope I pronounced that correctly.
That's correct.
Great. New York University, and Georgia Panagiotakos of the University of California at San Francisco. So welcome to you all. Thank you. And I thought I would start with you, Susanna. I'm going to ask each of you to take 2 to 3 minutes, describe your work, how you hope it contributes, and some challenges. So Susanna, if you would get us, you've got a clinical perspective, which is a wonderful place to start.
Great. Hi, my name is Susanna Mierau, and I'm a neuroscientist and neurologist at the University of Cambridge. I graduated from MIT as an undergraduate in 2000, course 9, and did my graduate work, my PhD at Oxford and my MD at Harvard. So in my clinical work, which was primarily done at the Massachusetts General Hospital Lurie Center for Autism, I saw children and adults with autism who had, as the diagnostic manual now defines autism, difficulty with social communication and interaction, and restricted patterns of behavior and other interests. But in the clinic, we really see autism as a very wide range, not just in severity, but in presentation, which suggests that autism is likely many disorders that are being grouped together. However, a growing number of genetic mutations are being identified. And so in the research work that I've done, both in Boston and now in the University of Cambridge, I've really focused on autistic syndromes that have a single gene mutation that's responsible for the problem. Basically, we want to understand how do these gene mutations disrupt the communication between brain cells? Because unlike in some, say, common forms of dementia, such as Alzheimer's, where a lot of the dysfunction is accompanied by cellular loss, or kind of loss of brain tissue as a whole. In autism, and especially in Rett syndrome, which is one of the syndromes that I study, the cells don't die, but they're clearly dysfunctioning. And so basically what I do is I record electrical activity from different types of brain cells in mice that have the same mutation as the children with Rett syndrome. So in Rett syndrome, the children, they totally look pretty normal for the first 6 to 18 months, but then they start to lose social, language, cognitive, and motor function, become very disabled. And it turns out when we make the same mutation in mice, the mice also look normal for the first few weeks with a much faster progression, and then they start to lose skills as well. And basically what I hope that these studies will tell us is, one, how does this mutation affect different types of cells and the communication between these cells and the nervous system? And also, since we find many of the effects of these mutations happen quite early in development, it should also help us determine when do we need to start treating autism if we do find kind of molecular targets that we can develop drugs to address.
Wonderful. Susanna, thank you. that loss of function, I know, is a real heartbreak in families, and it's very interesting that you're trying to key in as to why that might happen. Next, I'd like to, Jean, I'm wondering if you can tell us about your work, please.
Yeah, sure. Can you hear me?
Yes.
Yeah, so my lab's interested in studying the basis of how proteins cause issues in downstream molecular changes. And so we have two areas of research, actually. One area is in neurodegeneration, which, as you know, is an aging-related disease. And the other area is autism, which seems to be a developmental disease. And so we are interested in building methods and computational approaches to basically analyzing in a scalable, reproducible manner, molecular as well as cellular changes that are derived from cells, stem cells, or iPS cells from patients, and also cells from patients where we put in engineer-in mutations that are single point mutations of patients with autism, or also large-scale chromosomal amplifications and copy number changes. And so the hope is to basically derive a convergent signatures either at the molecular or cellular level that can reliably predict how a variant that causes autism or sort of eventually the phenotype, and to be able to then use these models as scalable systems to then do drug screens, to do chemical modifications, to try to reverse these aberrant molecular and cellular changes.
Thank you, Jean. I'll be interested in hearing maybe more, if we have time, about why you chose those specific genes. That's fascinating. And Neville.
Yes, hi, can you guys hear me?
Yes.
Okay, great. So I'm Neville Sanjana. I was both a PhD student at MIT with Sebastian Sung in brain and cognitive sciences. And I also did my postdoctoral work at MIT in the Broad Institute with Feng Zhang, also in brain and cognitive sciences. And so I came to New York just about 5 or 6 months ago and started my lab here at the New York Genome Center in NYU. And a lot of the work that we do here actually kind of leverages some of the technology that I developed as a postdoc at MIT. And so some of this is kind of similar to what you just heard from Jean, which is that we take advantage of two pretty recent technologies. One is genome editing, which is just the ability to write DNA, something that until very recently was very difficult in human cells, and combine that with stem cell technology to do something very unique, to take some of the mutations that we're finding through sequencing patient populations, kids who have autism, taking these mutations and in a high throughput way, being able to introduce them into human cortical neurons. And so without stem cells, it would be very, very hard to actually have that kind of nice model system. We can't, of course, take neurons out of human brains, but with stem cells, we can actually produce these in the lab. And so that's a very powerful technology to combine with the gene editing technology. So that's a very high level overview, and maybe that's enough to start with.
Super. And regarding how you hope it'll be of use.
Yeah, so maybe I can give you one more thought about that. So with this enormous increase in our ability to sequence genomes, what we've been able to do is discover all these new genes that we think might be implicated in autism. But right now, this is just correlations. We just observe kids who have some mutations in these genes. And at the end of the day, that's not very satisfying. What we'd like to do is actually put these mutations in and really understand in our own lab systems, what are the molecular mechanisms that might be driving the disease. So the idea is really to translate the reading DNA side of things by using a writing DNA technology to really understand what is the mechanism of the disease. Can we create a model system in the lab that will enable us to screen for therapeutics? That's
fascinating. Thank you.
Yeah.
And Georgia.
Hi.
Hi.
Can you hear me?
Yes.
Okay,
Thank
great.
you.
So I, like Susanna, was an undergraduate at MIT, and I continued on before graduate school, actually, in stem cell biology. I spent several years focused on the directed differentiation of immature pluripotent stem cells into specific kinds of neurons. And I went on to Stanford, where I complemented that background with mouse modeling for developmental disorders like autism. And like all three of the panelists, my work is heavily focused on trying to understand molecular mechanisms that are involved in driving changes in the brain during development that give rise to behaviors that we recognize as within the autism spectrum. In particular, like Susanna actually, we focused on types of autism that are caused by mutations in an individual gene. And the value for us of that kind of work is it enables us to kind of dig down and define more concretely how a single genetic mutation can give rise to a very clear change in a brain circuit and how that might give rise to a behavior. And the goal of my research at UCSF is to complement this kind of model work with stem cell-based models from my past, if you will, to really try to find what I like to call convergent cellular mechanisms. So there are so many genes that have been implicated in autism. And I think for many of us studying molecular mechanisms of the disorder, a key direction is going to be, can we identify similar cellular defects or similar molecular defects that all of these mutations happen to converge upon, or at least some fraction of these mutations happen to converge upon. So it's exciting for me to hear the work that my colleagues are doing. And I want to turn it back to you, Catherine, so we can get this going.
Thank you, Georgia. I think you're all brave souls to be wading into the complexity of the human brain. And it sounds like you're using the best and newest tools at your disposal to try to sort it out. Just to pull the camera back a little bit, a question that I think a lot of families are wonder about, or I think anyone who reads about autism may wonder about, is how does it start? Is it something that people are likely to inherit in their genome? Is it something, are there environmental factors in the womb? We know that the refrigerator mom model of autism causes long out of date, but might there be some exposure in the womb, or might there be exposures as something like a lot of families are concerned about in the home or in their communities? Or the work that you're doing, is it giving it any insight into that?
I might be able to go first on this question. So as mentioned by all the panelists, there's a big attention placed on genetic causes for autism, and that at least represent, probably a large section of the pool of people who carry an autism diagnosis. Part of this is that for the psychology or more cognitive science end, which we don't really represent here of research, is finding earlier and earlier, now even into the less than 6-month period, biomarkers to start diagnosing people with autism earlier. And along with the many mutations that have been found, I think suggests a strong genetic cause in which these changes may be happening already in the brain during development before the baby's born, but are definitely occurring afterwards too. There have been a number of other causes suggested. There's been a lot of people looking into interactions between the immune system and the nervous system and how that may contribute. It's not an area of my expertise.
Yes.
And of course, there may be environmental causes that are identified in the future. However, I think what we do know is that the environment is one way in which we can actually really improve the outcome of people with autism. For example, the earlier we give attention to children and start, applying therapies to help them learn social skills, help them develop language, the better they'll do later on. And we also suspect from some of the cellular and molecular work that we've been doing that when we identify drugs that can directly modify the brain development, that the earlier that we apply these, likely the better the outcome as well.
I think the main evidence of the reason I think so many of us are focused on genetic causes of autism is that for a long time, actually, even before kind of the modern genome sequencing age, it's been known that autism is very heritable, that if in twin studies, so if one genetically identical twin has autism, there's a high likelihood that the other twin will also. And so I think that's one of the primary reasons that there's been a last 15, 20 years of ramping up of investigations into the genetic basis of autism.
It's interesting. We have a question already from one of our listeners. It's from Prissa in New Hampshire. She asks, other than Rett syndrome, in these single genes that are implicated and chosen for study, do you know if the phenotype is robust, or is this not yet studied? Let's see if I can make sense of that. In these single genes that are implicated and chosen for study, do you know if the phenotype is robust, or is this not yet studied? Is that clear to you folks?
This is a great question. Yeah, I think I'll let maybe Jean should step in on this first, but I can say that in our work, the phenotype is one of the most difficult things when we're taking on a disease like autism, a social disorder, and then we're using cellular models, which are very powerful. We can do a lot of molecular assays. We can play with the genome in certain ways. Even with mouse models, it's really difficult to say what is the phenotype of autism when we really have it more in the sense of human children. So,
yes.
Yeah, so that's a great question. So I think in our research project, we have this grant from the National Institute of Mental Health, and it's a large consortium grant involving 5 different labs and 3 different companies. And the main purpose of the grant was that the recognition from NIMH that actually a lot of studies that are using sort of the same cellular models that we are using are sometimes hard to reproduce perhaps in an academic setting, going from line to line and so on. So in this project, we actually have about 12 to 13 syndromes which have a genetic basis, ranging from Rett syndrome to Fragile X to Phelan-McDermid, Williams-Barrett. And what we're doing is taking 1 or 2, sort of base lines, IPS lines from single individuals that are normal, and then using genome editing tools that Neville mentioned to put in this controllable way these variants, and then try to build really scalable and robust phenotyping measurements from neurons generated from these lines, from 3D mini brains generated from the lines, from looking at synaptic connectivity. And interestingly, we're trying to work with companies. So NIMH requires in this interesting grant that we have to build in the grant replication studies for any of the phenotyping assays that we have, and also that the companies that we're working with have to be able to translate the assays developed in the academic lab into a scale that really any company and any lab can run at, and so is are the phenotypes from Rett syndrome replicable? I think that yes, and I think that we'll see from our and other data that the other phenotypes should be robust as well. I think the key is to find the right sort of measurements. That's a longer answer to your question. Right?
No, that's super, and thank you to Prissa for that question. There's another question from Larry in Cambridge, and I'm not sure if it's the Cambridge near MIT or the other one. Do panelists have any hope for cord storage, and I assume that means umbilical blood cord storage, being a factor eventually in helping individuals overcome this disorder? I know I'm based in North Carolina. There's been a lot of research at Duke using cord blood treatment, not necessarily with stem cells, but with what I'm told are progenitor cells. So I don't know what you folks are, whether you see hope in this line of research. Okay, Larry.
Yeah,
I was going to say, Larry, we'll have to go elsewhere for that one, but it's certainly, you're correct, Larry. It is an active, at least where I live, it's a very active line of research. I'm sorry we couldn't.
So we see cord storage research as becoming more popular in the areas of degeneration, where where as folks age, they are losing neurons, or they're losing astrocytes. And then I think if you have cord blood stored before, technically you could, theoretically you could make the cell types to replace sort of the cells that you have lost. I think that is not, as far as I know, maybe the other panelists can tell me better, but I don't think this is a main area of research in autism.
Yeah, my sense is that this cell replacement has been investigated to a far greater degree for degenerative disorders where you get massive amounts of cell loss. But as Susanna was mentioning in the beginning, cell loss seems like less of an issue in a lot of these development, at least in the spectrum, so to speak. So
Yes.
the value could be more from a disease modeling perspective, right?
Mm-hmm.
But there, the induced pluripotent stem cell platform has really been, I think, the primary direction that a lot of people have moved into, where they can sort of take people with defined genetic backgrounds and turn their skin cells into these pluripotent stem cells and study phenotypes in a dish. But I've not heard too much about cord blood being used for that purpose.
Okay, another good question. Thank you, Larry. I'm going to interject here and ask one question I wonder about, which is from your work and the range of your work, do you folks have a sense of the variation among the biological basis for autism? We always hear about the spectrum. People present very differently. They have similar traits, lack of engagement with or motivation to be engaged socially, some communication problems, sometimes some repetitive behaviors. But people with autism on the spectrum, some have immense gifts, some are highly disabled. They're so different from one another. And as you guys focus where you're focusing, are you getting a sense that there is similarities across the spectrum? Or do you sort of hinted at this, that maybe it's actually a collection of different diseases?
Yeah, I don't like the name spectrum disorder because it implies that they're all like somehow variants of the same thing. Yeah, I mean, at the end, with more careful genomic research, it might end up being, hundreds of different, disorders with perhaps similar enough overlapping phenotypes, but they have different molecular bases, perhaps, and the way you would treat each of the genetic, each of these different genetic disorders might be a different drug, right? A different time point in development where you would treat the patient. So I don't know. That's just personal opinion.
I think one good analogy I'll just say is with cancer. There's something where the phenotype, the high level phenotype, is very similar, right? It's uncontrolled growth, and so with autism, we think the high level phenotype is this primarily a social interaction phenotype. But as we know that different cancers, whether it's like a blood cancer like leukemia or a brain cancer like glioblastoma, the genetic basis of these is very, very different. And so I think now that we're able to kind of peel back the layers of the onion and start doing genome sequencing on these folks, which has been a really well-funded effort over just the last few years, and still there's much more discovery left, primarily because the human genome is huge, 3 billion base pairs. I think that it's just going to be the case that we'll see an evolution like cancer, where separate diseases are really separated out.
Yeah. And the genetics seems to be rather complex, right? So there are some very rare mutations that are very penetrant that can cause, very clear cellular phenotypes. But there seems to be some evidence that there are common variants. So these are variations in the DNA that are common to more than 1% of the population, right? That have small effects in isolation, but perhaps there's additive effects of multiple mutations. And we still don't understand sort of which mutations contribute to which feature of the disorder as well. So there's a lot of emphasis trying to link specific mutations with distinct aspects of the disease, right? So like, is there something that's very specific for social defects or language impairments or the stereotypies that you see in the behavior?
Yes, and I was Susanna, yes, good, because you're from a clinician's point of view, I was hoping you'd weigh in.
Well, I agree with the other panelists that, I mean, the numbers of different genetic mutations that are going to be identified in patients, I think, may exceed the number of hundreds, or even once you consider, multiple mutations. I won't try to make an estimate on that. But I do think that one area of convergence, for at least from my research perspective, is for the neurobiological basis, is looking at the area of the synapse. And this is the area where neurons communicate with each other. And there have been a number of mutations, especially the highly penetrant ones, like the, like Rett syndrome, Angelman's, Fragile X, which have a very clear syndrome. Obviously there's variation within it, but the person who asked previously about how robust are these phenotypes, for a few of these syndromes, it's very robust. And then we also have a number of mutations identified in proteins at the synapse that have been identified in siblings with autism, where we don't know for certain that those mutations are causing their autism, but it's attractive to us because those mutations are in proteins that we know are critical for the way that neurons communicate together. So even though I think that we're going to find at the molecular level many different causes, we know, one, that the synapse is likely to be very important, but also two, on a kind of a clinical level, we know the different areas of the brain that are involved in social and communication. And so a lot of people do research at a kind of much more higher cognitive level, where they may be able to, even though there may be many different causes, they may be disrupting a similar system. So there may be the possibility for some common treatments in the future.
So it sounds like, as from a clinician's point of view, Susanna, you see grouping these people together, whether it be on a spectrum or some other way of thinking about it, it makes sense to treat them as a whole.
Well, especially since most of our treatments right now are educationally based, or working with this, very profound sensory deficits that people have. So at that kind of cognitive level, we find that, whether or not people have language problems for many different genetic reasons, they all benefit from working with speech and language therapists to improve. Now, in the clinic, it's fascinating because, the types of language deficits can be very different. Like, for example, some people, it's profoundly expressive. Like, they really can only produce a few words, or they're completely nonverbal. And that may give some people the impression that they don't understand what's going on. But when many of those individuals have later been taught to use assistive devices, it turns out that not only do they fully understand, but many of them will start typing in, complete, full grammatical English. And often too, even the ones who can speak, what's coming out doesn't match what they would like to say. For example, one of my mentors told me a patient who would always, just repeat the same phrase over and over. And then, I love you. And then she typed on her assistive device, I'm really, ticked off that you kept me waiting in the waiting room for over half an hour, and so, I mean, while as other people have very severe, what we call receptive language, where they don't really it's not just the social cues they're missing. So I think language therapy works for everyone, but it also has to be customized based on, the individual.
Thank you. Another question. This is from Amy in New York, wondering about what types of medicines are used. But I'm going to edit her question a tad. And if there are effective medicines out there, what are they? But what kind of therapies from the research that you folks are doing can you envision in the future that might come out of some of these genetic-based studies?
So I just say, in the clinic, currently all the medications we have are used to treat symptoms of the disease. We don't have any medications that that we think are actually altering the course. For example, many people with autism suffer from severe acid reflux. Many people with autism suffer from seizures. Many people have behavioral issues, which are so strong, like maybe they rub their hands together so much that the skin is coming away. We actually don't have very effective treatments for that, but we do try to, use the kind of psychiatrist's pharmacologic toolbox to kind of improve behavior. But I'll let everyone else comment on what type of future strategies might work, because we're quite hoping to have some.
You need them.
Honestly, I think the number of strategies probably scale with the the varying number of genetic causes, right? So one can imagine things like Fragile X or Rett's, where you have basically a loss of a function of one single gene. One can imagine things like perhaps gene therapy, where you are replacing this gene or editing the promoter, so you are allowing the gene to be expressed and produced. There are cases where you have a entire chunk of a chromosome missing. One would have to envision identifying maybe the key genes that could be replaced. Cases where the convergence is at the synapse. One may envision trying to build molecules to enhance the scaffolding of these proteins or the transmission or transmitter. So I think there's going to be a whole range of different drugs that one could develop. But the key is still, I think, the phenotype, and I think it's hard to develop all of these drugs. I think in a fast or efficient manner using mice models these days. I think one can hope that the IPS models provide a much faster way, once the phenotypes are robust and reproducible, right, to then identify these new treatments.
I agree with what Jean just said, and I think one of the key challenges, and this is, I think, one of the exciting things that we as scientists can work on right now with autism, is whether it's, Jean mentioned small molecule like drug therapies, or whether in some future scenario you can even envision some gene replacement therapy, something like that. Independent of what kind of biological method we're thinking about addressing these diseases with, I think a key question is, at what stage is the intervention going to be effective? So one thing that's kind of lacking in our knowledge right now is we don't know whether at a later stage, if you intervene at the time a child is 5 years old, 10 years old, maybe an adult, can you reverse some of these deficits? Is it possible for the brain to kind of, even at a later stage, be taken to back to whatever is judged by the clinician to be the desirable or normal state? And especially in cases where very severe forms of autism, where there's very low IQs, things where the parents really desperately want the condition to be addressed, but they have, a teenager. And I think it's a challenge, right? It's an open question whether models like IPS models or mouse models, induced pluripotent stem cell models or animal models can really help us get at this. I think it's something we haven't really tried rigorously yet, and it's something that's interesting, because even if the answer is no, then we know, okay, this intervention needs to be done really quite. So I think that's an exciting neuroscience question.
And there's a lot of evidence to suggest, actually, that a lot of the changes that you see in the brains of patients with autism actually emerge very early in development. So many of the genes that have been implicated seem to be very important during mid-gestation, right, which is really rather early. And we actually have some evidence using several of these genetic mouse models and sort of complementary human IPS models of some specific genetic forms of autism, that we get very similar defects in the kinds of neurons that are generated in the developing brain. And so I actually often wonder, where would intervention actually be best, right, in this context? Because if you're building a brain and you're not losing cells at a large scale, but what you're actually doing is sort of titrating the types of neurons that you're getting and how they connect to one another. Yeah. The synapse might be actually a really good place where you could target your therapeutics as the sort of output of all of this brain development. But I think this is going to be probably one of the most exciting areas, right? Can we identify some convergence from all of these genes that are coming out, and then target a very specific process using all the new technology that's coming out?
I think there's a related, very relevant question. Maybe I should have left this for Catherine to ask, that just popped up here on our screen, which is
go for it.
Barry from Florida.
Yes.
Mentions that often results in mice don't translate in clinical trials to humans. And our researchers studying this in primates, and it's not work I'm working on, but I can say that since I recently left MIT, there's two folks there, Guoping Feng and Feng Zhang, both in brain and cognitive sciences at MIT, I know are very interested in being able to use primate models, which just for this reason precisely. But I think Georgia and Jean probably will have maybe even some more insight on the role of mouse models versus primate models.
Mm-hmm.
Maybe.
Yeah, this is kind of a mouse. As it turns out, mice are pretty fundamentally different from humans. The value of the mouse, obviously, is they're genetically easy to manipulate, right? And you can study an intact circuit element. What's kind of lovely about this model is that it's relatively short, and you can watch cells divide and make these decisions that in the human are on the order of 9 months in something like 18 days. But what we do know about the human and the primate brain is that during the development of the brain, there's actually different classes of progenitor cells that emerge and different ones that exist that are maybe specialized. So for example, here at UCSF, a major discovery that came out about 5-6 years ago was a completely new class of progenitor cell called the outer radial glial cell that many people think might be important for expanding the upper cortical layers, which is something that's unique to primates and humans. So I think the IPS field, to some degree, is trying to complement what we see in mouse models and see if we can identify sort of places where the mouse models are valuable, i.e. intact circuits, and places where we really do need a primate model. If we want to study mid-gestation development in the human, I'm not sure that the mouse is going to be enough to see human-specific features of this disease, right?
Well, go ahead, Jean.
No, I was going to say there's one more thing to add. I think the non-human primates, provide a complementary model, really, because I think there's also, reliance vision for social signaling. There's all these different aspects of the behavior, social hierarchy that the non-human primates have that mice are maybe not, not sufficient. So, or rodent models are not sufficient. So I think the non-human primate research is really critical. Now, the only issue is always time, right? So it takes a long time for models to be developed in non-human primates and then to be evaluated and so on. So I think the IPS models, at least for the 3-dimensional models, are a good bridge, to try to speed up or at least funnel in high prioritized targets, and then one can then go to non-human primates with a lower risk of failure. Yeah.
And another listener has a question that might be, I don't know if this is related to the 3D modeling, Jean, but this person's wondering, TJ from Sudbury, Massachusetts, is wondering about the future of artificial intelligence and what it could do to help in the diagnosis and treatment of these kinds of disorders.
This is definitely a question for Jean.
Okay, well, I mean, you can look at it 2 different ways, right? One is, there's a lot of efforts in AI in terms of machine learning to be building up basically methods to analyze the imaging data from the basic sciences or the high throughput sequencing data to try to evaluate what are the key pathways. So that's one way. The other way, I think folks are trying to develop robotics, right, to be able to, like little robots to interact, with kids with autism. Actually, maybe Susanna has more to say on the clinical aspect, but there's some evidence that just having these robots there to interact with these kids in the long term may prove very valuable. But yeah, that's.
Yes, I think that key to improving social interactions to have social interaction. So I guess in some ways I'd probably preference human-to-human interaction. But there was a very cute story in the New York Times a few years ago about a woman wrote in about her autistic child's interaction with Siri. The response.
Yes. Lovely. Yes.
And it actually it may not have been a therapeutic intervention for the child, but it was a therapeutic intervention for the family, because a lot of autistic children like to ask the same question to
again and again,
greater repetitive frequency than their family can tolerate. And Siri is very tolerant, but also too when apparently sometimes with this child would ask things that were inappropriate, Siri is apparently, through the humor, I guess, of Apple programmers, does do a little behavioral modification. So they were also getting some input like, that's not an appropriate question to ask, or whatever. So I don't know that it, I think there could be much more sophisticated stuff, and people have actually been using machine learning to try to refine like which questions on the diagnostic, these 3-hour diagnostic tests are actually most predictive of autism, like as in, can we actually shorten it down to make so that we can have more? But I think there is potential there.
And it's great that Siri sometimes has a sense of humor. That helps things too. Well, gosh, we only have 3 minutes left, but I'm going to throw out a question to y'all to wrap up, which is, and it's too short, I apologize, but we don't have enough time probably to really expand upon it. But if asked, what do each of you consider, and why don't we go in the order that we started in, just what do you consider the biggest obstacles to the kind of insights you're really hunting for regarding these disorders?
Well, I think the biggest obstacles surround, and this addresses some of the questions that we weren't able to get to, just the kind of higher level, how are these higher level cognitive features really being produced? And, the genetic, whether we're using mice or stem cells to address it is, I think, a great place to start, because we can be confident, at least for those specific syndromes, that we're addressing the biological mechanisms. But somebody else asked a question about pesticides or, environmental influences. And, there are labs that are working on those causes, but, there's also the additional challenge, which I think is particularly, is that we now know that in some forms of intellectual disability, that you only need 3% of the cell, particular cell type in that area of the brain, to have a, mutation in order for the disease to be expressed. So, for example, after the brain, as the brain is forming and the cells are moving to their different areas and dividing, when they divide, they can get mutations. We call these somatic mutations. And just having a few cells misbehaving in the cortex is enough to disrupt it for the whole, that whole section of the brain. And if we draw blood or take skin cells to make, pluripotent stem cells and drive those, we're never going to capture those genes. Because those mutations are only happening in a very small percent, and very few people are, volunteering their brain for biopsy. So I just think
probably a good policy.
There may be many sources out there, that we can't really get a handle on right now. And so I think we're starting in a good place, but I do see some big hurdles.
How about, let's see, if we go in the order that we started in, Jean, in terms of what's the biggest obstacle in your work, would you say?
Well, I wouldn't. Okay, so there are other obstacles in my work that are more technical, but I think just fast forwarding to the future. Let's say we do have drugs that we can identify, that we can treat a patient, and ideally, a lot of these drugs are going to be used very early on. Maybe prenatal, right? So very early on, when the child is still in the womb, to affect a proper neurodevelopment. Well, a big obstacle there is, how do I know if the drug I'm using is working, right? Even in clinical trials, how do you know that? And I think, I don't have a solution, but I think that would be a big problem for many, many different diseases as well as autism.
And Neville?
Yeah, so I'm going to turn around the question a little bit. I think instead of talking about obstacles, I think what's interests me right now really is what we don't know. And so one of the things that our lab is focusing on here now at the New York Genome Center and NYU is really, what is the contribution not just of genes? We're getting an incredible resolution of what genes might be involved in autism. And people, other panelists have said this is a very long list of genes, but there's so much other stuff in the genome. There's so many of these what we call non-coding elements, the regulatory elements, or you could call it dark matter of the genome, something like that. This other stuff that is there, which is actually the majority of the genome. And I think I'm really curious to see how much of this, because there's a lot of missing heritability in autism. There's a lot of genetic sources of autism that aren't really explained just by looking at the loss of function of genes that we've considered so far. And so I think this is going to be a really exciting area for people doing the genetics, and hopefully for people like me who are really trying to understand what are the biological mechanisms underlying these mutations. Yeah. So.
And then, and forgive me because we're running out of time, but Georgia, can you give us a quick response?
Nice and quick. I'm just going to echo what all the panelists have said so far and agree that how the maternal environment actually interacts with a lot of the genes that have been identified, and whether you have additive effects of the genes and the environment is something that I think is particularly interesting, and will be an important challenge, but I think one that will keep us in business for a while as scientists for many years to come.
Many years, many decades. Well, I want to thank each of you and wish you the best on what sounds like very promising and difficult work. To our listeners, on behalf of the Alumni Association, thank you for tuning into this Faculty Forum Online Alumni Edition. This is Catherine Clabby. I'm signing off. Thanks again to our alums from UCSD, NYU, UCSF, and Cambridge. We'll be sure to pass all questions asked by viewers on to the panelists today. You can continue to chat about today's panel using the hashtag MIT Better let me start that again. Hashtag MIT Better World, which from my time at MIT, I know is a true commitment of that institution. And please send up any follow-up questions or feedback to alumnilearn@mit.edu. Thank you all.
Thank you.
Thank you, Catherine.
Thanks again for joining us. For more information on future MIT Alumni Association productions, please visit our website.